indicated, and many of you, a couple of you, hopefully all of you eventually have indicated an interest in a job. In my ongoing quest to make virology about more than viruses, I thought I'd point out there's a local company called Deerland Enzymes. I do some work with them. They're looking for a quality control technician, so it's kind of the entry-level foray into the science world. I'm sure it doesn't pay very well, but they do research and development, and there is an opportunity to move up. Some of my former students work there. They at least tell me they like it. So if you're interested, get in touch with me. I'm going to basically ask you for a resume that I can send to them. And they're within a couple of miles of campus here, so convenient to be a student and technician. You don't need a degree, but you need to be working toward one. So enrolled is okay. They prefer somebody with a degree, but QC technicians are hard to keep. Secondly, I wanted to draw your attention, if you're unaware, to our weekly molecular biosciences seminar series called MBIG. It's something I started a few years ago to create a real research seminar here. Sort of the first one is now being modeled in other disciplines. If you're interested, there's also EBOT, which is ecology, environmental biology, and organismal biology. So that's sort of the other side of molecular biosciences, which I imagine is what most people here are interested in. But the idea is to have a forum to communicate scientific results and tell people about your research. About half the talks are local people from KSU, and about half are outside speakers. So you can see we're having a big dog in a couple of weeks who's actually a famous geneticist, worm geneticist out at California, Berkeley. So the idea of doing this is several-fold. One, to communicate results. Two, give our researchers a forum, give our researchers the ability to learn cutting-edge results from elsewhere and in other fields, and to plug students into the research community. So many of you want to go to grad school. I know I've talked one-on-one with a lot of you who want undergraduate research projects. If you start, I would encourage you to come to these talks. So this week, actually the next 3 weeks, are all local KSU researchers. One is a grad student, two are faculty members. And start learning what's going on here, because that's going to help you plug into a research lab you're interested in. You may be interested in joining. It's also a good sign that you're actually interested in research, and so the people who want to take students will shop for them. It's kind of a dating market for mentors and research students. And it gives you the ability, if you want to start shopping around for grad school. So, I don't know, the worst fates than winding up in Berkeley, California for 6 years. You can start to get to know people and network through there. So that's weekly on Fridays at noon in Clendenin 1008. Please consider coming. Oh, and you can, not this semester unfortunately, take it for 1 credit hour. It's offered on a repeating basis. It's going to be regularized into the curriculum going forward, but right now it's just 1 hour of elective credit. If you want to do anything other than be interested, show up and not fall asleep in a puddle of your own drool. Another thing I'm in charge of is our college-wide science talk seminar series. You may have seen these last semester. We certainly had a couple of big ones. It's occasional, but coming up on February 4, we're having Dr. Manu Platt from Georgia Tech. We're trying to build a relationship with Georgia Tech, and he was excited. He helped us on a grant proposal, and while I had him on the phone, I suckered him into coming and giving a talk. And you can see he's quantifying proteolytic activity to predict disease potential. So he's biomedical engineering. If you're interested in things, I don't know, biomolecular, biomedical, this would be a good thing for you to come to. I'm going to also have him meet with students, so there'll be an opportunity for you to sign up to have a session with him to ask about not his research, Georgia Tech, grad school in general. He's a very positive, outgoing guy. It'll be a big experience for you. And there'll be another one in April. It's a classmate of mine from high school at Duke. We'll keep you informed as we go along. Oh. Now, many of you want to go to grad school, some of you are graduating this spring. It may be possible that you don't have anything to do come fall. And so I wanted to throw a possibility at you. I wouldn't encourage most people, but since I have something to do with it, I thought I'd at least mention it because the deadline happened, but we didn't get enough applications. The deadline has been extended, you can still apply. There's a master's in integrative biology here at KSU. It's now in its second year. It's a 2-year program. It's supported. You don't pay tuition. You get paid $12,000. So it's not good. Plus more in the summer, so it can be somewhere around $16,000 to $18,000. And the idea is to either get you a terminal master's degree for which you would need to get a job that needed a master's degree. My interest is in preparing people for doctoral programs. So if you don't get into a PhD program, it's sort of a diamond in the rough. We'll squeeze you into something a little higher purity and higher carat, to extend the analogy a little too far, and hopefully ship you off to the doctoral program. It's in general a bad idea to stay where you are. So I don't broadly encourage, like I say, I'm broadly encouraging this. It may be a decent option for you under certain circumstances, so please feel free to talk to me about it, and you're always free to go apply to the program as well. If you graduate with a biology degree, of course, you are qualified to go to the master's program. And lastly, a student has asked me to pass on to you that KSU is hosting the Science Bowl, which is, I believe, a high school science competition, and is in desperate need of volunteers this Saturday at the refreshing hour of 7 in the morning. If you're interested, good thing to show community engagement for those med school apps, talk to Dr. McElroy. So there has been some confusion, also, one other note before we start. There's been some confusion about what's due when. So I've been getting quizzes later than the deadline. So I would just, they need to be done by the deadline. So check the syllabus. I'm clearly not going to be delineating crisply between chapters. We're still in chapter 3, but the quiz for chapter 3 was due last week. Chapter 4 was due today. Chapter 5 was due Monday. It's basically a 1 chapter a day thing. But there's sometimes we'll touch on 2 chapters, and there are 2 quizzes you can take for that. But the syllabus has what the schedule is. Any variation from that, I'll let you know. But things are due. The class before we're scheduled to start talking about the class before the class in which we're scheduled to start talking about that subject material. Okay, so we had ended last time with the notion that viruses are macroscopically symmetrical. And I had asked why. I don't believe who it was, because they're small. They have small genomes. If you want a complex structure, I guess macromolecularly, I'm symmetrical as well, at least in some ways. Their genomes are small. They don't code for much more than a few proteins. So if you only have a few building blocks, you only have a few shapes. And if you need to enclose something, then that shape has to be regular and symmetrical. So why are viruses symmetrical? Because they have to be, because they're small. So we'll touch on 2 larger categories of shapes: icosahedrons and helical viruses. Now, any Dungeons and Dragons nerd can tell you what an icosahedron is. Or my Dungeons and Dragons nerds. Well, nobody played Dungeons and Dragons?
No, but it's,
A senior level science class, nobody played Dungeons?
no, but it's 20 sides.
You've played Dungeons?
No.
You know, in my youth, I played Dungeons and Dragons rather than talk to girls. Right. 20-sided figure. So there it is. So there's an icosahedron. And then helical, of course, are repeating symmetrical structures. Why does a helix make sense given, an icosahedron makes sense given a limited number of building blocks because the faces of the icosahedron are the same shapes, and icosahedrons give you an efficient volume as opposed to a cube, or a more spherical shape. Or you can make spherical shapes from icosahedrons. You can make efficient volumes to enclose the nucleic acids inside. Helical viruses are another way to be symmetrical with a limited number of building blocks. Think about DNA for a moment. So there are 4 nucleotides in DNA, and it forms a double helix. But really, all it is stepping through space while stepping through an XY circle while translating in the z-axis. So if I had a spiral staircase here, we're going up, I'd be going up a helix. And so we can have one basic shape and the right geometry to create the helix. So it's a very simple shape and encloses nucleic acids that are long polymers as well. So let's go to back to icosahedrons for a moment. These figures, the figures from your text are okay, but I like these better. So the link is there. This is not up, this current version is not up on the site. I'll try to replace the one there and get you the link. So we have a 20-face shape that this is of course great simplicity. In reality can be spherical, but you can look at the underlying macromolecular geometry and notice that there are indeed 20 faces of regular repeating structures. The icosahedron has 2, 3, and 5-fold axes of symmetry. That means I can run through the structure with an axis and then rotate around it, or I can do it with 3 axes and rotate around it, or 5. That does not mean that any face of the icosahedron is composed of one protein. So that was back, Watson and Crick actually did a lot of the early viral structural work, and they thought, well, if you need 20 faces, given the genomes of viruses, you should have things working, and the virus they were working on, should have things in multiples of 60, but that wasn't the case. That wasn't the case because most viruses will have multiple proteins that form structural units of the face of the capsid. And so we can take a real virus, look at the global icosahedral shape, but realize that there are faces composed, these are oligomers of a hexamer of 2 different subtypes. So there are 6 proteins that are forming one part, and then there are pentameric shapes, pentamers, so there are oligomers of proteins that have 5 repeating subunits. And thus we have hexamers and pentamers going in to make the faces of the icosahedron that then have the overall global shape. So we can have a simpler example where we have 3 subunits on the face. Here where we have 6 plus 6 plus 3 or 9, making up one of the faces. And again, the icosahedral geometry of the pentamers and hexamers does not have to make a 20-sided die like 20-faced triangular figure. It can be spherical because of the pentamers and hexamers that uses the analogy of a football. I was thinking for a moment, by the way, do I give credit for the student who points out that that's not football? My standing offer on LCA points, if you find an error in the textbook, you get 5. The first to post it, you get 5 points. But then I looked, and the author's already British, and it was published in Spain, so technically it would not have been an error, but fortunately nobody mentioned that was a football. And so hexamers, of course, have, this is a hexagon, hexameric shapes have 6 intersecting faces. And then we have the pentamers, which in this example here are blue. But the soccer ball is spherical. It has an icosahedral geometry, so geometry and shape are not the same thing. And of course, looking at the gallery of viruses from your textbook, you can see all these viruses have icosahedral geometry. They have very different, well, for viruses, very different structures. The viruses themselves. Of course, these are all false colored to enhance your view of the subunits, and they don't really look like this. They really look like this, and so that's an electron micrograph of a different virus than this. This is papilloma virus, but structural subunits that are discrete are called capsomeres. So this is a capsomere. However, it still is the icosahedral geometry. You can see that that's a hexameric shape. Yes, a hexameric shape. Oh, I didn't wear my Carolina shirt. Although this is from my high school. It's the North Carolina School of Science and Mathematics. We were the unicorns. I was on the wrestling team. Tobacco Road, North Carolina, 2A high school. We'd have to go do the wrestling against the Harnett County Trojans. We pranced in the gym. And the worst part is, so the school used to not sell stuff with the logo on it, because it's just for nerds, and they didn't want to emphasize athletics. But then the budget started getting cut, so we had our unicorn was like My Little Pony, very frilly mane. That didn't sell very well, so they ripped off the Denver Broncos. So now we have a badass unicorn. It's the Denver Bronco. That doesn't have anything to do with virus. I'm sorry, capsomere. Yeah, capsomere. Just move on. You know, my biochem classes, which I can sort of teach in my sleep, this is a very new experience for me if you can't tell, have standing orders whenever I just go off on a tangent, don't come back to yell squirrel. I don't know if you've seen Up. The dogs always get distracted by squirrels. So I know that. Yeah. So please feel free to yell squirrel. And so the capsomere is discrete shapes. Knobs and fibers are types of protein found at vertices. I want to say vertexes. And often have a role, as you would expect, in attachment. So we have a fiber here on the cold virus. That fiber is a projecting protein that is part of the pentamer, so it's a penton, projecting a role in attachment. So beyond the simple icosahedral geometry, there are in many viruses structural or proteins that play roles other than merely enclosing the genome. Of course, it has to get in someone else, or it has to be able to infect its host. And then a lot of bacterial viruses will have tails.
You said these help with host attachment?
Was that you, Corey?
No, that's me. You said that helps with host attachment. Did you say if it helps with like breaching?
I did not say that. I did say the first thing. The roles of pentons will vary by virus and by its life cycle, and we are going to go through in excruciating detail in the middle part of the course different viruses and talk about their life cycles. For now, you can imagine there are other roles for pentons and hexons. You can imagine that this plays a role in attachment because it's projecting forth from the structure. The virus has to have a productive collision with a host. It's like Velcro. The projections from Velcro, they help the sides attach.
Gotcha.
I was actually giving a talk at a Sunday school class a few months ago about Myers-Briggs personality tests. I don't know if you've ever taken them. This guy came in 5 minutes late, sat down and interrupted me and said, "Did you talk about how the NFL uses this?" I haven't gotten around to that. You had to be, never mind. Tails. I didn't say the second thing, but it was a good question. Bacteriophage tend to have projections and tails that also play a role in attachment. And then bacteriophage, because bacteria do not do endocytosis. So we'll see in the next chapter, a cold virus will adhere to a host cell and then be endocytosed. Clathrin-coated pits and all that, we'll talk about that in detail. Bacteria don't do that. They don't have subcellular compartmentation, so the phage has to inject its genome into the host, has to attach. So these projected structures, these structures of the tail allow for attachment and injection of its double-stranded DNA into the host.
So it's called a tail, but it's really more like a syringe. Right?
So, I don't know. And I don't know that we know. I do know that it's an active area of investigation. How do bacteriophage get their genome in? It's not like a syringe in that you push a syringe. So what's pushing the DNA? Nothing. Could passive diffusion explain it? I don't know. I have a friend at Michigan State who works on a phage of Shigella, and actually we're collaborating. It's called a spike in the Shigella virus, and we're actually just looking at the basic protein-protein interaction between the hosts. We have no idea how injection occurs in this Shigella virus. It may be known for T7. T7 is one of the oldest, one of the most well-studied bacteriophages because it's an E. coli phage, and E. coli is one of the most well-studied bacteria. And then finishing up chapter 3, finishing up chapter 3, some viruses are enveloped. Here's one of your classic figures for your textbook. Some enveloped viruses have spheres. They're spheres. Some are bullet-shaped, like rabies. So what you're seeing here, if you're confused, you can refer to the color code, is lipid envelopes, of course, are host-derived, and then proteins projecting. And so those different shapes are a result of the capsids of the protein and then the lipids enveloping them. There are examples of proteins enveloping the lipid layer in other viruses as well, and then they have different shapes, which we'll talk about when we get there. And then just to lastly mention, another viral structure is the occlusion body. Some viruses, particularly insect viruses, pack in arrays that allow them to survive, called occlusion bodies, that allow them to survive once they've killed the host in the environment and persist. So we'll see an example from chapter 4 of baculovirus, which infects insects. These occlusion bodies come out of the host and then they sit on a leaf and wait to be eaten by the insect. So it's sort of the tick, the viral equivalent of a tick. A tick can sit on a leaf for like 3 years waiting for something to walk underneath. By the way, do you know what a tick senses? Butyric acid, which is a component in sweat. Ticks have two senses, right? They can smell butyric acid somehow. I don't really know how tick noses work. And then temperature. So when you walk past that plant, the tick just lets go because it smells your sweat. Speaking of sweat, Suzanne. No, I'm sorry.
Do they act like the spores found in bacteria?
Yes, they're the viral equivalent.
Okay.
The spore being the spore equivalent of a tick.
Okay.
So a lot of bacteria will form spores that allow them, what does that allow them to do? It allows them to persist in the environment. So sporulation is often a response to stress. So if bacteria that form spores find themselves in an environment lacking in metabolites, they'll sporulate and wait for the environment to get better. You can think of an occlusion body as, oh, the virus is thinking, of course viruses don't think. The virus thinking, oh shit, my environment just got bad, I killed. Oh no, sorry. My environment just got bad, I killed my host. Better form an occlusion body and wait for a new host to eat me. Sorry.
So is it one virus or a group of viruses?
It is a group of virus, commonly insect viruses, because transmission from host to host involves more time and space than does, say, flu virus, which goes from host to host in droplets of moisture. So it's the viruses that tend to have to persist in their environment and do not have, excellent transition to chapter 4, a vector. So let's start with some global points here. So viruses have to go from host to host. Why?
Because they're small. They can't live without a host.
Because they're small, they're parasites, they're non-living, they can't replicate on their own, they will kill the host and then need a new host. So a virus that doesn't transmit its progeny is a virus that does not exist. But viruses are really bad at what they do. In some ways they're great at what they do, but they're really bad because viral transmission is a very rare event. What has to happen for a virus to transmit? It has to emerge from one host, it has to go to another host, and it has to have a productive collision with the host cell. So a lot of the viruses we'll talk about are transmitted by sneezing and coughing and breathing in. Think about coming from an endothelial cell into a spit droplet, going to another host, and then productively causing an infection. It's a rare thing. So I just said viruses were bad at that, but why might that be a good thing? From the virus's point of view, sorry. It's a good thing for our point of view.
Because eventually they'll like if they were really good at it and it wasn't so rare, then they'd outgrow their resources.
Being really good at being a parasite means not killing your host. And that's why we don't really have to worry about World War Z. That sort of transmission and infection, if it occurs, tends to attenuate. So is anybody reading the 1918 book?
I will be when Barnes & Noble delivers it.
Barnes & Noble still exists?
Yeah.
downloaded it.
Yeah, so one of the take-home points of that book is how infectious that flu virus was. And, you know, it showed how 50,000 people in Philadelphia died in 4 weeks when the city was much smaller than it is now. But what happened? It attenuated. It became less infectious. And if you cause your host to go extinct, you go extinct as a parasite. So it's a good thing from the viral biology point of view that it's a fairly rare event. And what do you care from a viral point of view? You're using all your host's metabolic energy to replicate. So, I forgot to say this. Most viruses never fulfill their special purpose of infecting the host. But that's okay because they're using the host's energy to produce them. Let's cheap copies. Sorry, I'm questioning my ordering of things here. We want to just introducing more vocabulary. We have vertical and horizontal transmission. Vertical is from one generation to another, perhaps through the progeny are infected by their parental, transmission occurs from parent to progeny. And a horizontal transmission is between individuals. And this can be through a vector or host to host. Which begs the question, what's a vector? Mosquitoes are vectors. Circular definition. Viruses are transmitted through vectors. Vectors are organisms that transmit the virus. Often it's an organism in which a virus can persist, but does not parasitize. So one example is West Nile virus. That's of interest here in the U.S. It's a pretty pathetic virus in terms of most people who get it never even know they have it. It's pretty bad at causing infections. However, it does cause encephalitis, so when you get it, if you get an infection, and if it's a bad one and you don't react well and treat it, you might die. Around 1,200 people have in the U.S. But mosquito is an example of that because it can, in biting a host, inoculate itself with the virus that's in the host. The virus persists in the gut, works its way back to the salivary glands, in the neck. When the mosquito next bites a host, the virus is then transmitted into the host body. They are frequently arthropods, insects in our case. And some vectors are circulative, which means the virus has to circulate within the mosquito. So the mosquito eats the virus, but then the virus has to come, according to my salivary glands, if they were mosquito-like, has to move from the gut to the salivary glands. It has to circulate within the mosquito. I don't really know why I picked West Nile, other than I lived in Connecticut at the time West Nile came to the U.S. So this is a virus that emerged in Uganda in 1937. It was never a problem in the Western Hemisphere until a decade or so ago. It first emerged in New York City in, I believe it was 1999, and has spread throughout the United States. And you can see it. It's not quite a pandemic. So these are incidences of cases of West Nile in humans. The darker it gets, the more the more infections there were in that area. It's a little bit deceiving because those areas are bigger than these. As an aside, if you want to study infectious disease as a physician, the place to do it is New York City. So the first case of West Nile was in New York. My wife went to med school at a place called SUNY Downstate. It's attached to one of the worst hospitals in the country. It's a great place to get a medical education, if I should use that phrase, because it's a terrible hospital. And they don't have enough doctors, so they throw clipboards at her and tell her to go see patients, which is patently illegal. If you go somewhere like Emory, all the patients there have private insurance, and they know enough to tell the people with the short coats not to touch them. But so she was at Kings County Hospital is the name of it, and it's a terrible hospital for anything except trauma and infectious disease. And it's because of the population. It's in Brooklyn. They see a lot of diseases because of people coming in and out from all over the world and living in high densities. It's a great place for infectious disease to break out. And so we see transmission across humans in the country. Why do you think you see so much in the Great Plains?
You said it's like per 100,000 people, and it's not that many people.
Yeah. So that explains the area on the map, but not the color.
Do they have less access to like hospital care systems? Or
I hadn't
is
thought
it in
of
the
that.
middle of nowhere?
They don't have any medical schools. So if you're from North Dakota, you can get in-state tuition at Washington State. Not helpful, I know. Amber.
I know, like in New York, they spray to kill the mosquitoes
Yes. So
everywhere.
this is a huge thing in New York and Connecticut was do they spray because nobody wants to be sprayed. They sprayed because everybody panicked about it, and it turns out it didn't turn into the epidemic. Every new viral outbreak is going to be the thing that turns us into World War Z. And it didn't. You can see it's receding. The answer is because there are a lot of livestock there, and West Nile has more than one mammalian host. It's perfectly capable of infecting cattle as well as people. Incidentally, it's a dead-end virus. It doesn't go from people to the person-to-person transmission. I don't think has ever happened. It persists in birds. So mosquitoes bite birds and then bite people. Neither here nor there. Anyway. So one aside here, so viruses infect all of the domains of life. Plant viruses are different. I'm going to write it. Plant viruses have a problem. Transmission across, you got to get across the cell wall to infect plant. And so they are a big barrier. As with other viruses, we see insects as the vectors. Insects eat the leaves. But when they eat the leaves, they don't disrupt cell walls of living cells, right? They eat the cells that they eat. They tend not to damage the other ones. So speculated that plant viruses get in through damaged but retained cells. Just to show you a plant virus, tobacco rattle virus infects, it's a counterexample, of more than 400 species. This is corky ring spot, so when it gets in potatoes, you can imagine what this does, what this could do to Idaho. Everybody's going to want that potato. And it goes through via a nematode. I hate plants, or enough about plants. I don't hate plants. I don't like studying plants. Don't worry about photosynthesis. Vertebrate viral transmissions, usually we're going from epithelial cells in upper respiratory tract, coughing, sneezing, inhalation. We'll talk about the genital secretion, well, actually we will. Genital secretions, GI shedding, so hepatitis virus, for example, is shed through poo. So the gross factors here is fecal oral. Fecal oral transmission happens far more frequently than you'd like to think it does. And through lesions such as warts and sores, and of course, bites. This is rabies virus. It's the bullet-shaped one. And so I thought I'd take a few moments out to bring your attention back by showing you some good stuff. So, a little animation of the transmission of rabies followed by a clinical example.
Spread through the saliva of infected animals like dogs that affects the central nervous system. Once the virus enters the brain, it causes acute encephalitis, inflammation of the brain. The disease is characterized by aggressiveness, extreme thirst, excessive salivation, and extreme paralysis, leading to death.
That sounds pretty bad. So we have sort of a novel mode of transmission here. Sorry, don't know what these are coming in. We have sort of novel mode of transmission here. We're not coughing and sneezing. Lots of viruses that go that way. It's not fecal oral transfer. It's saliva from a puncture wound going into the blood, causing encephalitis, which is bad. Lots of other things too, but rabid dogs are crazy. It's an interesting case of a virus affecting behavior, and that behavior resulting in transmission of the virus. So it's a co-evolutionary cycle. Because of an encephalitis, the animals are more likely to bite the future host of the virus. So the virus causes the behavior that leads to its transmission.
Are people infected with rabies more likely to bite people?
Yes. Didn't you see the skin? The dude? People, yes, they are. I'll show you that in a second. I'm going to ask you a question about rabies on your first test. You're not actually going to have to know anything about rabies beyond what you already know. It's going to be a big think question. So this is something, as those of you going to medical school, will never see. So we know there are, I can't remember the number, there's been something like 39 deaths from rabies in the U.S. in the last 30 or 40 years. There are cases. The reason why is there's a very effective treatment for it. And so to bring you an example, we have to go back to like 1930s rural Iran.
See this because I try to explain to people exactly what you'll be looking for, what it'll look like if people get rabies, and so it's probably best that you guys see it with your own eyes.
This is a couple minutes, but it's really.
I mean, I had to watch it so you guys can watch it too.
Old American Society for Microbiology film.
This group of villagers from the backcountry of Iran were all attacked and bitten by a rabid wolf. They have come to a local facility of the Pasteur Institute in order to receive treatment. This man developed symptoms of rabies. On the third day of the disease, he displays a characteristic restlessness and agitation. The most familiar clinical symptom of rabies is associated with difficulty in swallowing. When fluid comes in contact with the throat, it is violently expelled, at the same time producing painful, spasmodic muscular contractions, hence the term hydrophobia, or fear of water. The accessory muscles of respiration are also affected by the attempt to swallow, so that the patient chokes and gasps for air. In order to avoid swallowing, he drools accumulated saliva into a cup. By the fifth day, salivation is copious and almost continuous. There is excessive perspiration, and the pupils are dilated. As a precaution against the convulsions which commonly occur in rabies, the patient is secured hand and foot. He displays increasing agitation and delirium. If the acute excitement phase of rabies is survived, paralysis sets in, accompanied by apathy and stupor. The patient becomes quieter. He lapses into a coma, which is followed inevitably by death.
How do you like the doctor with bare hands? That's not standard of care today. Here's the weird thing, is we haven't done that. It's available on YouTube clearly. 1937, Iran, or making up the 37, long ago, rural Iran, to get, there are actually other videos available of people with rabies. Why don't we see, why are you, those of you who are going to be physicians, probably never going to see a clinical case of rabies? Caveat, with the additional information that when a successful infection is established, the clinical course of the disease would still be the same today. You wouldn't, as a physician, you wouldn't be able to stop it. What? I don't mean to see human cases. We do. I mean, there are some.
Because we're vaccinated.
We have an effective treatment. There's a vaccine. Why aren't you vaccinated?
Because everybody's been vaccinated.
No, nobody's been vaccinated with rabies. It's not standard of care to give a rabies vaccination.
They give it to all the pets, right?
Yeah, dogs and, but why don't people? So they also give it, by the way, to veterinarians and people who come into contact with animals that are rabies vectors. Why? Pretty horrible. We're around animals. Why aren't we? Yes.
Because it's not very effective, Andrew. Because it's not very effective in humans.
It's completely effective.
No, because I'm
Oh,
vaccinated.
you're talking about memory.
Yeah.
I'm sorry, I do know what you're
Yeah,
saying.
you have to get rabies titers every 5 years. And you also, if you actually are bitten, you still have to go back and get a booster.
Okay. Thanks. How do you know so much about?
Because I'm a veterinary technician.
Good to have the information. Thank you. Let me see how to incorporate that into the point I wanted to make now that I have new information. The same is true of other things for which we get vaccinated. I mean, there's a very real world concern here as to why we don't get vaccinated for rabies. That would be one of them. Another one is, it's not often you get bit by a rabid animal, right? The post-bite treatment is effective. There's the worry about side effects of vaccination, but the real reason why is it's expensive. So there's an effective treatment that is cost effective because very few people ever get it, where the same is not true of, say, a measles vaccine. None of that, by the way, is what I'm going to ask you. My point here is vertebrate viruses are transmitted mainly via arthropods. Of course, there are physical, inanimate vectors as well, such as contaminated needles. Mainly blood-sucking insects. Viremia is the presence of virus in blood products, or in blood. Presence of virus in blood products makes presence of virus in blood has historically been a medical problem for drug use, and also just wanted to make a point about science, why we're so great. And in hemophiliacs, so a genetic disorder results in blood clotting defects in either factor 8 or factor 9. So when you get a cut, blood coagulation, I used to study blood coagulation back in the olden days. By the way, just squirrel thing here. If you walk into your first graduate lab and your advisor says, I want you to go kill a cat, you say, okay, I'll loan you my bow and arrow. Leave that lab and go to another one. So my first project as a graduate student was cloning a gene involved in the blood coagulation cascade from cats. And he wanted me to go steal some little girl's cat, kill it, take its liver, and make a library, a clonal library from the liver. I eventually wound up getting a cat from the pathology department. But blood coagulation is a very highly regulated process, as you can well imagine. If you get a cut, you want to form a clot at the incidence of the cut, but not in the lung. That's bad, or the brain, or other tissues. But the problem is, of course, that your blood is circulating. And so blood coagulation is very closely regulated, and it's a cascade of proteases that are named in the order in which they were discovered. Blood clotting factor 1, factor 2. Factor 2 is thrombin that actually is the initiating of the clot formation. These other proteases involved, in which 8 and 9 are critical, not critical, are very important, are circulating coagulation factors. And what happens is there's an initiating event, and proteolysis starts happening at the site of injury, but as the blood flows away, there are other proteases that shut off, that cut up the proteases so that you form a local clot. Hemophiliacs have genetic defects. There are any number of them in factor 8 or factor 9. When they get a cut, they have to get functional factor 8 and factor 9. And back in the olden days, the 50s, 60s, and 70s, what they would get is chilled plasma from another person that would contain functional coagulation factors. Actually, I was reading about this last night. Hemophiliac children would lie to their parents about getting cut because getting pumped with a large volume of cold serum from someone else is not a pleasant experience. It required a hospitalization. And so then they'd die. They would hide their wounds from their parents. But after the next technological development was a concentrated serum produced by freezing serum, so that you would get concentrated coagulation factors. And this may guess where I'm going. With the outbreak of HIV, the first populations affected were homosexuals and hemophiliacs, because they were getting viral containing clotting factor concentrates that contain viral particles. And so this was one of the first big wins for molecular biology. So since the late 80s, recombinant factor 8 and factor 9, where the brand names is Benefix, have been available. It's a hard thing to make a blood coagulation factor because they have a really weird amino acid. So there are 20 amino acids. Glutamic acid is one of them. So that's normally glutamic. That's normal glutamic acid. There's an enzyme, which actually was the gene I was trying to clone from a cat because somebody in the Netherlands had a house cat that was defective in the enzyme, which is the vitamin K dependent gamma glutamyl carboxylase. My job was to go figure out what the defect was. And this a lot, so it's post-translationally modified to gamma glutamyl, gamma carboxyglutamate, or it's glutamyl when it's in a protein. That allows it to bind calcium, which is necessary for its activity. Bacteria don't do that. So when we think about recombinant proteins, at least when I think about recombinant proteins, I think about taking a gene, sticking it in a plasmid, putting it in a bacteria, making a bunch of it. Well, bacteria don't have the enzyme that's required for this, and that's why clotting factor concentrates persisted for hemophiliacs long beyond where you thought they would. It was a matter of producing functional coagulation factors. You needed humans to do it. Benefix is actually made in CHO cells, so Chinese hamster ovary cells. It was a big victory for molecular medicine. And just last year was approved for prophylaxis. So what hemophiliacs would get injections of clotting factor concentrates when they had an injury. Now it's just last summer approved for regular dosing in order to prevent bleeding recurrence. Transmission of invertebrate viruses, the distinction is that invertebrates are not only the vector but also the host. So this is an example of a caterpillar, died from baculovirus. Those are the inclusion body. Occlusion bodies we talked about earlier, they persist on a leaf until eaten. They're packed in a crystalline array. It's in the gut of the worm that the individual virions break free and cause infection. So this is the example of the occlusion bodies allowing the virus to survive long enough to be eaten by another caterpillar that comes along after the one dies. And then in order to transmit, the virus has to replicate. In order to replicate, it has to reach a permissive cell. Global theme that virology isn't hard. A permissive cell is one which allows which permits the virus to replicate. So many viruses are tissue specific. Hepatitis, for example, infects liver cells. It has to get to the liver cells in order to replicate. And that's often protein-protein contacts on tissue-specific cell surface proteins that allow attachment. It's often the type of cell because the virus must be present. Some, for example, require mitosis because they require host enzymes that are only synthesized at that part of the cell cycle. So coming full circle, this is the life cycle of the West Nile virus. It does not require mitosis. It is a single-stranded RNA virus, so we have an enveloped icosahedral shape, attachment to a host cell receptor, endocytosis into an endosome, escape from that endosome into the cytoplasm, what's called uncoding the genome coming out of the capsid. We then have single-stranded RNA that is effectively a messenger RNA that encodes viral proteins and allow for eventual packaging of new capsids. So the nucleus does not dissolve, or it's not necessary that the cell go through mitosis because it doesn't need nuclear proteins. It can be packaged directly. The RNA encodes via translation the enzymes it needs to replicate its genome and then be packaged in new virions and exocytosed, with perhaps cell lysis being the exocytosis role, but it's actively transported back to the membrane. The host cell has defenses. We mentioned very briefly the exotic CRISPR-Cas sequence a week or so ago. Bacteria have restriction enzymes. So bacteriophages are largely double-stranded DNA, maybe exclusively double-stranded DNA. Bacteria have enzymes. You have probably, anybody who's taken biotech, actually everybody should have used these. Did you do the restriction digest in genetics?
Yeah.
So restriction enzymes hydrolyze the phosphodiester bond in a specific sequence. Usually they are palindromic, meaning they read forward and backward in the same sequence. That's because the restriction enzymes are dimers. They have to cut two phosphodiester bonds to break a strand of DNA. They exist to cut viral DNA that's injected from that phage. They don't cut the cell's own DNA because bacteria also have methylases that methylate their own DNA that prevents the restriction enzyme from productively, i.e. catalytically, binding to the DNA. So when you do molecular biology in bacteria, you always have to worry about whether your DNA is methylated or not. And effectively, you don't have to worry about it because laboratory strains are null for methylases. depending on the organism. Eukaryotic cells are another matter. Your textbook mentions that there are programmed responses to double-stranded RNA. This is not something, dsRNA, this is not something that's normally present in a eukaryotic cell. And so it's an effective thing to target against, a host cell defense. And so backwards. So this is just an example I pulled from the literature. I will put the link to the paper if you're interested. I'll put the link to the paper on the website in case, in detail, in case you're interested. So double-stranded RNA in a eukaryotic cell is met by host defenses, which are binding proteins and transcriptional response, which is also a function of binding. So in this example, PKR, can't remember what PKR stands for, but binding to double-stranded RNA activates PKR that then goes and phosphorylates a transcription factor. Sorry, no, that's not a transcription factor. An interferon that, when phosphorylated, binds in the ribosome and inhibits protein synthesis. It also activates another pathway that destroys RNA. And so it's the specificity of the double-stranded RNA sequence that leads to a protein nucleic acid interactions that have a protein level and a transcription level or transcript level response to destroy the viral RNA and to arrest protein synthesis so that the virus can't make new proteins to make capsids to package its RNA. So it destroys on two different levels. And there are also single-stranded RNA viruses. There are single-stranded responses as well. RIG, which I believe is named that way because it used to stand, back when it was discovered, for really interesting gene. But this is a protein, so it's not italicized. Binds the RNA, and then via interaction with another protein is, sorry, this is non-immune response, turns on transcription factors that it's not that interesting to me. I had it down to notes. Yes, I did have a question. Non-immune cells have a transcriptional response that destroys the RNA. Immune cells endocytose the RNA, and we have a receptor response, so this is toll-like receptor, that turns on not only a cellular response, but an inflammatory response that leads to an immune system response. So if the immune cells are more than that cell level, because they're turning on an immune response that has an inflammatory response tissue-wide or organism-wide. This is on a single cell basis to destroy the virus within the cell. And so I hate to end early because I'm going to give you tuition work, but please forgive me. I'll take 5 minutes of your life some other time.